This PowerPoint presentation is on Lab 8, Carbohydrate Catabolism, specifically the experiment on cellular respiration by Anthony Santos and Madeline Kestenbaun. When a seed, such as the seed of a pea, germinates into a seedling, energy is required. In order to meet this energy requirement, seeds store carbohydrates as a source of energy. These carbohydrates are catabolized, and the energy release drives the production of ATP. In this experiment, we shall measure the rate of cellular respiration in peas by measuring the amount of CO2 they produce. We will measure 3 different kinds of peas. One group is germinating, another group is not germinating, and a third is germinating, but we boiled it in water previously. Why are we measuring CO2? Well, in the citric acid cycle, CO2 is produced as acetyl is oxidized. Our hypothesis is that the germinating peas will demonstrate the highest rate of cellular respiration. When the carbonic acid from the jar is added to the phenol red, it will turn yellow. So there won't be a reaction from the ungerminated peas or the boiled peas. Part 1. Assembly and incubation. On your bench there are 2 bottles labeled germinated and ungerminated. Fit the rubber stopper with the attached glass tubes into the respiration bottles. Then submerge the rubber tubing into the water-filled test tubes at the bench. Then plug in the rubber stoppers on the top of the tubing and take note of the time. Incubate for 60 minutes. Part 2. Measuring the rate of respiration. As stated earlier, we are measuring the rate of respiration as a function of CO2 gas production. It is difficult to measure the volume of gas produced because it is colorless and odorless. To overcome this obstacle, we will bubble CO2 through water, which produces carbonic acid, namely HTCO3. We will then use a pH indicator to directly measure the amount of H2CO3 in our jars. In this manner, we will indirectly measure the amount of CO2 produced in these reactions. First, replace the water in each test tube with phenol red. Resubmerge the rubber tubing below the surface of the liquid. Otherwise, the CO2 production would be pointless, as it won't be in the water. Then, remove the rubber stopper from the top of the jar and slowly pour water into the jar. Carbonic acid will be displaced and move through the rubber tubing into the tube filled with phenol red. A yellow color is an indication of acidic conditions, namely the large amount of CO2 moving in. Here's our setup. This demonstration how setting up. Here's the respiration bottles, here's the rubber tubing, and the test tubes. Phenol red is in there. We haven't poured it in yet. Ungerminated over here, germinated in the middle over here, and the boiled germinated all the way on the right. This video is one we did of the germinated peas. No reaction at first. Note that there's yellow liquid bubbling over here. Now it has a brighter, almost orange texture, or color I should say. So a reaction occurred in terminated peas. Sorry, having a bit of a typo here. Anyway, unterminated. That's it over here.
Sometimes some of these videos, I don't know if it's the shape or what's going on, but it's difficult to view.
No reaction so far. And still no reaction. No reaction for unterminated. And finally, the boiled peas. Sorry, the video is kind of sideways. But you can still kind of see what's going on. They're pouring the water in over here. Here's the tube. And still no reaction. And that one is there. So no reaction. Here's a picture of the end results. Reaction terminated, no reaction in boiled or unterminated. Just as our hypothesis stated. The terminated non-boiled peas were the only ones that reacted because they had the ideal conditions for cellular respiration. Right temperature, and they were starting to release lots of CO2. The unterminated, they weren't in the stage in which they had to undergo a lot of cellular respiration. And boiled peas, I'm not sure if you heard this before, but there are some enzymes that cause cellular respiration, so the increased temperature, it distorts the appearance of the enzymes so that they can't work. And that is all.